I am going to talk about several areas today. All of them are going to come back to childhood health and function. Unlike the speakers this morning, my focus and my sort of direction are very different, and hopefully you'll see that I'm on the same page as these guys, just coming at it from a different angle. So I know I'm preaching to the choir a little bit, but we know that kids are becoming more sedentary. It's not just the United States, it's all around the world, but this is definitely a problem that we've had to deal with. I have a colleague at UCLA, and he likes to say that the reason we're becoming more sedentary is that we're victims of our own ingenuity. We're too smart for our own good. We've engineered physical activity out of our lives. And there's a lot of truth to that. But what we know is that along with being sedentary is this prevalence of being overweight and unfit. And I'm going to leave it at that because I know I'm speaking to the choir when it comes to that. But having said that, what you might not have thought a bit about is our own national security. This isn't publicized as much. It is in the news a bit, but it's not as publicized as much as this whole childhood obesity epidemic. But obviously, as we know, overweight children become overweight adults in most cases. And this has implications for our own security. The U.S. Department of Defense estimates about a third of their military-age youth are ineligible for service due to their weight. They spend a lot of money on this, upwards of $1 billion. And there are many that are just flat out too overweight to serve. And so certainly this is a growing issue in a lot of different sectors. Much like obesity, childhood inactivity often leads to adult inactivity. Kids tend to sample physical activities when they're young, and they pick and choose, and then they take them with them throughout the course of their life. And how many of you are playing a sport that you played when you were a child, right? You played youth baseball, maybe as an adult you played in adult softball leagues, right? I played hockey as a kid, and I still play hockey today. I'm 40 years old, still playing hockey. And it's because I learned to play as a kid. Now, I have friends who learned to play it at 35, 36 years old, and they will never be able to be good hockey players. It's just not, it's not in them, right? And so it's kind of like learning to swim as an adult, right? It's not an easy road. What's alarming, though, is that this generation of kids it's thought through various statistical analyses that they're going to live less healthy and have shorter lives than their parents, marking the first time in U.S. history that this trend has occurred. So they're not just overweight and sedentary, but there's implications for a shorter lifespan and a less healthy lifespan. And to make matters worse, as we know, we heard about this morning, is that schools which reach 55 million kids per year are making matters worse. They're not making matters better, right? They're eliminating physical activity from the school day in favor of teaching formal academic topics. The problem with that is that when you look at the whole range of research out there, you look at the extant literature, and to look at this relationship between physical activity and scholastic achievement, the data go in the opposite direction. There are two basic scenarios out there. One is that physical activity is related to better scholastic achievement, and the other is that physical activity has no relationship with scholastic achievement, in which case you've built a healthier, more physically active individual at no cost to education. And so it's not clear why these policies are in place and why we're taking away from physical activity other than common misconceptions. Now, data from our own laboratory support this. These are data in about 260 kids. And let me just say at the onset, I'm going to talk about a lot of data today. But I'm not going to talk about sort of all the things that go into it, like demographics and health factors and things like that. I can tell you right now that when we do cross-sectional work, we spend a tremendous amount of time matching our samples. When we do longitudinal work, we spend a tremendous amount of time making sure that we've characterized our samples properly. So if you have questions about that, you can certainly ask me during question and answer. But if you just sort of want to gloss over it, you can assume that we've done our background work. And so these are about 260 kids. They're in 3rd through 5th grade. We went into the schools along with my colleague Darla Castelli, and we gave kids the FitnessGram test. The FitnessGram test consists of a pacer test, which is a cardiorespiratory fitness test, a field test for aerobic fitness. They get push-ups and sit-ups and a sit and reach test for hamstring flexibility and body mass index. And what you see on this graph here is you see along the x-axis the number of times they fell in the healthy fitness zone, the number of tests they passed in their PE class, number of FitnessGram tests they passed. Along the y-axis, you see the total score from the Illinois Standardized Achievement Test. This test, here we have just mathematics and reading. The scale goes from 200 to 400. And as you can see, there's a nice relationship between the more physical fitness tests passed, the better they performed. This is a correlational relationship. When we dug a little deeper, we found that it was actually the aerobic capacity test, the pacer test, that was related to this, that was sort of underlying this relationship, such that the more laps they ran, the better they performed on math and reading. Muscle strength, muscle flexibility were unrelated, and body mass index had a small but negative relationship, such that an increase in BMI was related to a decrease in test performance. Now, there's been a lot of labs who've looked at that sort of data. My lab went in that direction after spending a good deal of time looking at sort of underlying mechanisms. I'm coming from the opposite direction. My training is in cognitive neuroscience, and so I started to ask basic questions about brain and behavior and eventually extrapolate out to more translational aspects, and I'll finish with those at the end of the talk. Most of the field focuses on cognitive aging. Sort of the vast majority, since going back to really the 70s, and there's not a lot of us, but they asked all these questions about people who are 60 years old and over. And so back in the 60s and 70s when they did this work, they called them older adults. Now they're probably closer to middle age. But what we're talking about is a whole wealth of information looking at changes in cognition, changes in underlying brain structure and function that accompany this aging process. And they find that physical activity influences cognition, brain, and behavior. Now, a meta-analysis suggests that this relationship is both general and selective. That is to say that physical activity is generally related to improvements in cognitive function, but it's selectively and disproportionately greater for tasks or task components that require executive control or cognitive control, and I'll use those two terms together. So what is cognitive control? Well, a lot of people have a lot of different definitions of it. One accepted definition is that cognitive control is goal-directed behavior that uses subordinate processes in order to achieve some aim or some desire. It's comprised of three different aspects of cognition. The first is inhibition, which is your ability to ignore distractions, stay focused, the ability to override a prepotent response and instead favor a less potent response. Working memory, holding information in our mind, manipulating it, and then using it at a later time. And cognitive flexibility, which we oftentimes call multitasking, right? You drive it in your car, you have to look at street signs, you have to gauge the traffic in front of you, you have to, maybe ignore your kids who are in the back talking, and so forth, right? Executive control is used throughout our daily lives. We use it all the time. We're using it right now. Probably the best example I can give you of me using executive control is we recently refinished our basement because I have a 10-year-old and we didn't want to put anything really nice down there. We went off to IKEA and bought sort of like middle of the line furniture and got this piece of furniture home, opened the box, looked at that one little sheet of paper, and and knew there's no chance, right, of making it look like that. And so I basically took the picture, and I had to sort of make it mix and match and make it look like it was, and not have too many pieces of hardware left over at the end, right? And so that's executive function. So in the laboratory, we have a whole number of assays to use executive to get kids to elicit their executive function. Adults too. And so this is a really common task in my laboratory called a flanker task. It's conceptually simplistic, but it's actually quite a rich paradigm. And you'll see it a couple times throughout this talk. In the most classic example, you have kids looking at arrows. You can see in the top slide, the top one and the very bottom one, the arrows all point in the same direction. The two middle slides, they point in, the ones on the outside, the flanking arrows, point in an opposite direction from the center arrow. The task is really simple. Look at the center arrow, the target arrow, and press the button based on which direction the arrow is pointing. If the arrow is pointing to the right, press the right button. If the arrow is pointing to the left, press the left button. And in the first and the fourth slides, you can see that the flanking arrows are congruent with the target arrow. And so it's a pretty simple task. In the second and the third trials there, you can see that they point in the opposite direction. These are incongruent trials. And you have to override the prepotent response you have or inhibit looking at those flanking arrows and instead focus in on that target arrow and press a button. Through 50 years of research, we know that everyone in the incongruent condition responds more slowly and less accurately when compared to the congruent condition. So it is a good test of inhibition. Here's data with kids that are 9 and 10 years old. These kids, there are two groups of kids. They're higher fit and lower fit. Higher fit means that they were in the top 25% for their age and sex based on normative standards. The lower fit kids are in the bottom 30%. There's about 20 per group. And what we did is we gave them a flanker task while in an MRI, and we imaged their basal ganglia. The reason we image their basal ganglia is because this subcortical structure has been shown to be involved in cognitive control. And so what's really nice about it is that not all the basal ganglia is involved in cognitive control, only certain parts of it. So as you can see, the caudate nucleus, the putamen, and the globus pallidus, these regions are involved in cognitive control, and we find that there's greater volume in higher fit compared to lower fit kids. But the nucleus accumbens is another area of the basal ganglia, and this area is not involved in cognitive control. Instead, this area is involved in things like affect and reward, and we find no relationship. So we find that higher fit kids have more volume, only in select areas of the basal ganglia, those areas that support this cognitive task. And what I'm not showing you here is that they also perform better. I'm going to show you that a ton of times in the next couple slides. So we're finding that there's differences in brain that reflect differences in behavior as a function of fitness. Now, most of the work in my laboratory involves event-related brain potentials. We have fMRI data. I'm not showing them today because the slides get real messy. They're not just simple throwing brains up in a slide. But the ERP data is really where I was trained and been doing this work for about 20 years now. And the idea is that this is an EEG trace. And rather than just sort of looking at spectral EEG, what you have here is you have this cap on people's heads. We fill the cap with gel. We use high density, as you can see in the ugly-looking kid in orange there. That's my son. You can see that that all these different electrodes are filled with gel. There's 64 of them on their head. And we give them stimuli. And following each stimulus, which is the event, we get a response, as you can see under the trials that drop below it. So let's see if I can. These here are individual trials. This is the average. And the idea is that they get these different stimuli, and we get these known responses right here, these trials here, and then they get averaged together. And random noise averages out over the course of a number of trials, and we're left with these known signals, this trial average. And then we're able to decipher different aspects of this signal and make reasonable assumptions about cognitive functions. The reason I like this measure better than functional MRI is because functional MRI is limited by its system. Its system that it measures is blood flow. Blood flow takes about a second to develop. ERPs, they are on the order of a millisecond. We can identify 3 different cognitive functions that occur in the first 50 milliseconds. And so all of these measures have their place. The temporal aspects of the ERP measure allow me to get at very select aspects of cognition that happen in a very brief period of time. And so you can ignore the text. Basically, you can focus on that circle at the bottom, and that is our P3 component. P stands for positive. Positive is down in ERPs, just to make things difficult. And the idea is that this component has a number of different things about it. The first is the size of the component relates to the allocation of attention. How much attentional resources you're allocating externally towards the stimulus in your environment. So if you're sitting there in the most classic of paradigms called an oddball task, it goes something like this. Beep, 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 boop, beep, 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 beep. And when you see that beep, when you hear that beep, you get this little P3 each time. But each time it goes, it gets a little bit smaller because it's just the same thing over and over. But then you get that boop. And that one oddball engenders a very large P3 because you're sitting there, you're looking at stimuli or listening to stimuli in your environment in that particular scenario, and something changes. And when something changes, you upregulate your attentional resources, say, what's different in my environment, and you register it and you move on. You update your working memory and you move on. And so when you get that oddball, you get that large P3. And the second part about this is the latency. How long does it take to develop? Well, Quite simply, the longer it takes to develop, the slower you process the information. We want to process information quickly. We want faster P3s. So we have a measure of attention allocation and a measure of processing speed. So the first thing I'm going to show you is a cross-sectional study. We take our flanker test. I make it more difficult. The first time through in the compatible case, it's just like I explained it before. You look at that middle arrow, you press the button, corresponds to the middle arrow. The button, if the arrow's to the right, you press the right. If the arrow's to the left, you press the left. They do that 200 times. Then we mix it up. The second time through, we layer what's called assignment task on top of it, and we ask them to respond opposite to the arrow. So they still look at that center target arrow. When the arrow points to the right, they make a left-handed response. And so we've layered in a response-compatible, incompatible array or task on top of the array differences. We have higher and lower fit kids, about 25 per group, and we found 3 differences between higher and lower fit kids. Lower fit kids have a very small P3. That's what you see here in the blue. The P3 here is very small. It doesn't modulate. So they're allocating very few attentional resources, and they're not modulating based on the difficulty of the task, as you can see here. Higher fit kids have a larger P3 amplitude across both conditions, as you can see, and it gets disproportionately larger when the task gets more difficult. So right away, what we're talking about is that higher fit kids have greater allocation of attentional resources than lower fit kids, and they're able to upregulate even more when the task gets more difficult. Then the last thing you can see kind of here, but it's there, is that they're processing the information more quickly. The P3 occurs earlier. So they're processing more quickly, they're allocating more attentional resources, and they're more flexible in the allocation of the resources than their lower fit counterparts. We'll ignore off to the left. I'll come back to that later. Just looking at the behavior on the right, I would argue that higher fit kids not only have differences in brain, but they have differences in behavior. You can see when you look at the compatible condition, they're significantly different from the lower fit kids. They're performing more accurately. When we upregulate the demands of the task requiring even more inhibition, that difference gets larger. And you can see that even though the task gets more difficult, they're able to maintain their level of accuracy. Where lower fit kids, their accuracy decreases from one task to the next. And so we're showing differences in both brain and behavior cross-sectionally. Now, this takes us to what I've been doing for the last 5 years. For the last 5 years, I've been overseeing the FITKids randomized controlled trial, the Fitness Improves Thinking randomized controlled trial. This takes place at the University of Illinois with surrounding communities. We send buses to schools. They pick up kids after school, and they come to our lab, and they come to a gym next to our lab. We train them every day after school. We have them for 2 hours. This is based on the CATCH curriculum, but it's modified from there. Before they get to do that, they come to the lab on 5 days, 5 different days throughout the summer. We test them. We randomize them. Half the kids go to this after-school program. The other half go to a weightless control group. If they fall in the weightless control, the next year they can join our program. What we know is that each day they get approximately 70 minutes or more of intermittent, moderate to vigorous physical activity. We measure that with heart rate monitors, pedometers, things like that. And the kids come to our program every day that school meets for a full day, so about 150 of the 170 days of school in Illinois. There's efficacy in the program, as we can see from these graphs off to the right. If you just focus on the bottom graph, you can see that kids in the after-school intervention, they increase their aerobic capacity by almost 6% using a VO2 max test, which is pictured to the left. Weightless control kids are less than 1%. And so this VO2 max test is basically a gold standard measure for aerobic fitness. They run on a treadmill. If any of you have had your sort of cardiac test, I think men are 45, women are 50, to look at heart function. It's that sort of test, except we also pinch off the nose, stuff a tube in their mouth that they have to breathe in and out through. It's a real pleasant test. And then we run them until exhaustion, right? We run them until they can't run anymore. And that's considered the gold standard test. And it's a very good test to measure cardiorespiratory fitness. So if we look at the data, we used a flanker task first. We modified it with fish. And you look at the graph to the right, you can see that from when we looked at the post-test performance, we subtracted away the pre-test performance. Those in the intervention group improved by almost 8%. Those in the waitlist control group improved by less than 4%. So we saw almost a 2-fold increase in performance in those that went through the after-school intervention. We used another task called a switch task. This task is really difficult. They had to make judgments about shape. Was it circle or square? Judgments about color, is it green or blue? And then once they were trained on that, we threw it back and forth, and based on the hand direction, they made one judgment or another. And so this is a switch task that requires them to do multiple different things at once. They have to inhibit the previous condition. They have to activate the next condition. They have to hold both rule sets in working memory. And they have to flexibly switch between them. It's quite difficult. And same sort of thing, though. We find that kids in the intervention group improve by 10% from pre-test to post-test, and kids in the waitlist control group improve by less than 6%. So we're seeing differences as a function of performance in this, or as a function of time spent in this after-school program. When we look at the brain activity, look at just the blue, the colored ones first. What you see there is these are the same sorts of things. They're changes in brain activity from post-test minus the pre-test. So these are differences since baseline. And what you can see is that in just the most difficult conditions of that of those tasks, the incongruent condition of the flanker, or the one where they have to switch between two different rule sets in this switch task, we see that those in the intervention group had an increase in activation. That activation that you're seeing plotted there is the P3 component. I'm not showing it to you as a waveform. I'm showing it to you as a function of all 64 electrodes. And so what you can see is that red splotch there is the increase in the allocation of potential resources. You're seeing that increase in attention during task performance, and you're seeing improvements in the task as I just showed you. When you look at the control group, the waitlist group off to the right, you don't see the same amount of activation. They're not able to upregulate their P3 component as strongly. The other four heads are just images of which of the electrodes on the scalp were significantly different as a function of the statistical test we performed. And again, you can see it favors strongly the intervention group. Now, what's more interesting is that we looked at attendance rate, which is a good measure. I mean, kids have to attend in order to receive the benefits of a program. We looked at attendance rate, and what we found is that those who attended more had larger P3 amplitude, greater change in P3 amplitude, and a greater change in P3 latency. That is to say, the more they attended, the more their P3 amplitude was increased, and the more their P3 latency was decreased. Larger increases in attention, and larger increases also in the speed of processing. And we also showed that the more they attended, the better they performed on that switch task as well. So the greater the change in accuracy. And so we're seeing that there might be some dose-response relationship here as a function of time spent in the program. Now, I'm going to switch gears again, or maybe switch gears for the first time here. You'll find I'm a little bit schizophrenic. I talk about a lot of different areas as quickly as I can in my lab. I have 5 doctoral students that are all working in different projects because I have a lot of different interests. But I'm really passionate about this acute exercise piece. I think that it's really hard, as you guys know, and you guys tell me probably a lot more than I can tell you about this, it's really hard to create behavioral change. We don't ask people to change. We either force them to in our program, right? They have to be. If they're in our program, they're moving, or they go away. But you guys have, you don't have quite that control. It's not a research study, and so, you have to try and find ways to get them to change. Well, I think it's a lot easier to get people to change in small doses. And so one thing we've done is we've looked at what are the effects of one single bout of exercise. There are 20 kids here. These kids came to the lab on a couple days. On one day, they simply sat quietly for 20 minutes. On another day, they walked on a treadmill for 20 minutes at a rate of 60% of their maximum capacity. This is a moderately paced walk. There's no whining, no complaining. No one couldn't finish. They didn't break a sweat. It's just a moderate walk for 20 minutes. We counterbalanced the 2 days across the sample. And what we found was that following the days in which they walked, as you can see, this is a flanker task also. So as you can see here, this is when they walked. We saw an upregulation of P3 amplitude, especially in the incongruent condition, the condition that required the greater amount of inhibitory control. And so that's what I'm talking about right over here. Following the day they rested, you can see it's smaller. So we already found our upregulation of P3 amplitude. You can look at the waveforms and see that there's also faster P3 latency following exercise. We look at behavior. Reaction time doesn't change, but accuracy does. They become more accurate in that incongruent condition, the harder condition, following the single bout of exercise as opposed to following the day of rest. That increase is about 8%, 9%, which is very large for this task. We followed this up. We repeated it. It worked again. We did it with ADHD kids. It worked again. And here's data following that task. And so what happened is they exercised or they rested. We got the electrode cap on their head. We gave them the flanker task. We got the cap off their head. Now we're about 35 minutes after the bout of exercise. We gave them the wide range achievement test of math, spelling, and reading. And what you can see is that following that single bout of exercise, kids' performance improved on both reading and arithmetic relative to the day in which they rested. And the change in reading is, according to the manufacturer, a full grade level increase in performance. Now, I oftentimes get the question, would this work in schools? I can't tell you from my own experience, but I can tell you that others have been able to show you that. And I'll show you one graph of that in just a second. Let me show you one other graph one other figure before I turn to that data. And that is, are there individual differences in this? So we know it works for ADHD kids the same way it works for typical kids. But how does it work for kids who differ in their cognitive controllability? We all have different amounts of cognitive controllability, and they progress at differently throughout the course of maturation. And so when we did this test, this single bout of exercise, and looked at this test again in kids who are either high inhibitory controllers or low inhibitory controllers, we found that kids who are low inhibitory controllers actually improve to the amount to that equal of the high performers, where at rest you can see the high performers have a greater amplitude than the low performers. So if you look at the top, the greater this is the high performers and the low performers, and following exercise, there's no difference. So we're seeing a disproportionately larger increase for those who are low performers. When we look at behavior, we see that both high and low performers show the same relationship for P3 latency. Both of them have faster latency, faster cognitive processing speed. And we look at the measure on the right is not sort of the same measure I've been showing you. It's one of interference control, so lower is better. It's an accuracy measure where lower is better. And what you can see is that the lower performers actually show this large drop in this interference control measure, meaning that they're actually performing better. And so what we're seeing is that even in the lower performing kids, we're getting the same effects, if not larger. And then, as I said before, there are a number of people who've tried these sorts of tasks or this sort of application of this task in schools. Matt Mahar at East Carolina, he uses the Energizers break, which is a 10-minute break in the classroom, and he shows that there's substantial increases in performance following this Energizers break in terms of time on task. It's a measure of attention relative to the group that received another intervention. And so what he's showing is that in the schools, this sort of effect may be found as well. The kids are paying attention better. They're engaged more as opposed to some other sort of intervention. Now, I have some data on adiposity. I'll tell you that my lab has only been doing this line of work for about 4 or 5 years now. And really, the reason we got into it is because we were, when I started to run this FITKids trial 6 years ago, we were alarmed when we figured out that kids in East Central Illinois, that 60% of our sample was overweight or obese, and 66% fell in the bottom 3% for fitness. So this is, we didn't go recruiting this. Champaign-Urbana is a small town. We took anyone and everyone we could that fit our criteria to be in the study, and this was just what we got. And so we started to look at, and along the same time, I have a new postdoc, he's not so new anymore, but his interests were in nutritional sciences, and he's sort of a nutritional neuroscientist, sort of a new group of nutritionists coming out. And we started to look together on this idea of adiposity and brain health and cognition. I think you guys have seen these graphs a lot more than me. These are adults. The only point I want to make here is that we're not moving in the right direction. Decade by decade, we are becoming more overweight as time goes on. And here are data from 124 kids that were in the first couple waves of the FITKids trial. What you have along the x-axis is you have DEXA data These are data that we took a region of interest measure between L2 and L4, and we looked at central adiposity, so the amount of fat mass in the belly, and we compared it with performance on a couple different cognitive tasks. What you have on the y-axis is cost. And so on the left is inhibitory cost, on the right is working memory cost. So a higher number means more cost, worse performance. And what we see is that increases in central adiposity are related to increases in the cost of inhibition and working memory. They're performing more poorly. When we look at academic achievement using that wide range achievement test, we see a pretty similar relationship, such as an increase in central adiposity is related to a decrease in performance on these tasks. Now, I have other data. I'm going to show you those in just a second, but I want to stop here and just simply say I am by no means saying that this is a causal relationship, that increase in adiposity is related to a decrease in cognition. We don't know this to be true. I can't take overweight kids. I can take overweight kids if I had the support to do so and lean them out and look at differences in cognition. But it's not ethical for me to take lean kids and increase their body mass and then look at cognition. So we don't know the direction of causality here. We don't know if there's causality at all, and it could be due to some third-party variable, right? So for instance, we know that kids with a lower executive function also tend to be more overweight because they have a harder time inhibiting their intake. And so this is simply a interesting relationship at this point. And so here's another task. After seeing that initial data, we went out and we sought out a sample of 36 obese children and matched them with lean kids. And we gave them a standard Go task, which is that oddball task before. So it's tiger, 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 lion, tiger, tiger, tiger. And then we reversed it and gave them a No-Go task, where they pressed a button every time they saw a tiger, and they inhibited a response every time they saw the lion. And so that No-Go task is an inhibitory task that requires you to inhibit a response that we've built up. And what we find is that healthy weight kids don't differ from obese kids on the Go task. But on the No-Go task, we see that while both groups do more poorly, as you'd expect, it's a larger inhibitory task, that the obese kids perform worse than the lean kids. When we look at activation, this is the same P3 component, but this task elicits it more frontally. And what we see is that healthy weight kids have a larger P3 component compared to obese kids. That is to say that they're activating more of that component, they're allocating more attentional resources, and they're performing better. In a follow-up to that, we looked at one additional measure that I'll only briefly touch upon here. It's a different group of kids, and we gave them a flanker task. And what we did is we looked at this other ERP component, which is basically a measure. Back in 1990, when it was first developed, they called it the oh shit potential. Others called it the Homer Simpson potential because it was like, doh, every time he made a mistake. But basically what happens is that this is your conflict monitoring, your fact-checking, your sort of supervisor that's sitting on your shoulder. Every time you make a mistake, you have some system that tells you made a mistake. The best example I can give you is that if you're sitting at your desk typing away and you're sort of zoning out and typing, you know when you make a keystroke error. Because something in your brain tells you, hey, you just made an error. Because you have something that's sitting there going along and checking your performance. And that something's called your anterior cingulate cortex. It's the dorsal portion of the ACC. And there's a potential that arises out of that piece of tissue. And what we know is that every time you make a mistake, you have larger ERNs, larger error-related negativities, than when you perform correctly. And what we find is that healthy weight kids have larger ERNs than obese kids. And so you can see that large blue peak is the healthy weight, and the orange peak are the obese kids. And why does this matter? When we look at performance on the very next trial, we see that healthy weight kids perform better than overweight kids. And so when you make this mistake and this evaluative system kicks in and says, you made an error, another part of your brain, the dorsal outer prefrontal cortex, says, don't worry about it. I'm going to regulate this. We're going to exert more top-down control. And what you'd expect to see is on the very next trial that you act correctly, if it's possible. And so we've seen this since the 60s. And so what we see here is that healthy weight kids not only evaluate more strongly their errors, but they also tend to regulate better in that their performance is improved on the next trial. So I'll give you this here. This is a sidebar. What about our pets? You know, I figured you guys all know about humans and kids and all that, so I threw this in just to give you a curveball. Well, the problem is not just here with children. The problem is also here with our pets. It's very interesting, right? Yeah, and these are, although, this is how it goes, right? But it's not just our pets. It's all these other species. There's actually 8 other species that inhabit human-influenced environments that are also showing increases in weight gain. But it's not that simple. It's not just that they're getting richer off our own refuse, right, as this inner city rat is, because when we look at these species, it's possible that they're also experiencing their own adiposity issues, right, their own obesity epidemics. And what's interesting is that even when you look at laboratory animals, which for many, many decades have had the same basic feeding patterns and husbandry and all that, and that hasn't changed, they're also gaining weight. And so it is not as simple as or it's not as simple as the complex issue we make it out to be, right? It appears to be that it is a much deeper issue. And if you read out there are actually other papers out there that suggest that obesity might be due to some unknown virus or infection that we're not aware of. I don't know that I buy that, but it's out there. It's out there in the literature. But it's sort of an interesting sidebar, I think. So I'll return to things that more of what I know about, and that's memory. One whole other area that I'm familiar with that we spend a lot of time studying is memory. The reason we study memory is because there's great animal models out there. We know that the hippocampus is involved in memory. We know that the hippocampus is a piece of tissue in the brain that grows even during adulthood. So you can increase the volume of your hippocampus during adulthood many different ways. Some of it's proper nutrition, but others of it is through exercise. Rats that run more on a wheel than other rats have larger hippocampal volume. Well, we're wondering what the human analog of this is. And so we ran a couple of cross-sectional studies. This one's very different than all my others. We asked kids to look at pictures of faces and houses. And the first time through, they just simply had to decide whether or not it was male or female, whether or not it was the inside or outside view of a house. Then they got a 5-minute break. Then we asked them to do it again. The second time we asked them to do it, we asked them to make a relationship between them. We asked, is this person an inhabitant or visitor? Do they live in this house or are they visiting this house? And we didn't care what criteria they used. And believe me, kids have really wild criteria. But great, that's fine. They went through and they did it. And then they went through and they saw another set of 40 pictures, and they had to decide whether or not they'd seen these together before. So no, yes, yes, right? And so the idea is were they able to recall that these occurred? And the reason we asked that is because in the second case, when they had to form a relationship between them, that brings the hippocampus online. One of the things the hippocampus does is it creates associations. When that mouse is out foraging around the woods, it has to remember where the food is. It has to remember where it's secured, where its hole is to survive. It has to remember where the predators are. And it creates associations between these items and its environment. So when we look at the data, what do you know? In that single task where they had to decide male, female, inside, outside view, no differences. But when we brought the hippocampus online, we get this nice effect with higher fit kids outperforming lower fit kids. So we did this task a second time. We used pipes instead of faces and places, just to make sure that the people didn't look like someone's grandma, or the house wasn't something like a house down the street from them. And so they made decisions about whether or not these pipes occurred together or whether or not they occurred as a function of independently. And we found that in the MRI, higher fit kids had greater hippocampal volume. They had larger volume in that hippocampus bilaterally than lower fit kids. And we found that they also outperformed the lower fit kids on that relational accuracy task, not in the item accuracy task. And we found that hippocampal volume mediated that relationship between fitness and performance. So it appears that we're also seeing the same effect in humans. Now, I'm going to skip this one and talk about this task today. This one today, actually, I've been getting phone calls this morning from various news agencies because this paper just got put out yesterday. And so this is written up in Science Daily today, I just found out. This is we ask kids, so everything I've talked to you about before, up till now, is about performance. How well do kids perform? How will they perform on an inhibition task? How will they perform on a working memory task? How will they perform on scholastic achievement tests? We want to know, do kids actually learn better if they're more fit as opposed to being less fit? And so we gave kids these maps. They came in, this is all done on an iPad, and in one condition they sat there and studied the map. This fictitious map had these names pop up on it. They would see it in the study-only condition, and they'd press it to say they had registered it, they had seen it. In the test study condition, it appeared off to the side, and they had to tap where they thought the name matched the region. They did this 6 times, and then on the next day, they came back 24 hours later. They did a recall test. In the free recall, they just saw it, like you can see it there, and they had to type in the name in each box. And the Acute recall, they had it along the bottom there. They had this word bank, and they just clicked and dragged into these different boxes. Now, at the end of the learning day, it was pretty straightforward. Both groups, higher and lower fit kids, were equally successful, about 90%. So they went through it 6 times. They learned it over the course of those 6 times, and they were about 90% accurate, leaving the lab on that day. They went home for 24 hours. They came back, and we find that higher fit kids outperformed the lower fit kids on the study-only condition, where the test study condition showed no relationship. Now, why might that be? In the test study condition, we gave them a strategy. We allowed them to test themselves each time. And so it upped the performance on that task. But when left to their own devices and just told to study, it appears that only the higher fit kids, the higher fit kids perform better than the lower fit kids. When we matched it up by recall, you can see that was the free recall condition, even the more difficult condition, that differentiated these 2 groups. And so learning appears to have occurred at a higher rate in the higher fit compared to the lower fit kids under the most difficult of circumstances, when it was a study condition, was a study-only condition, and when they were required to recall it freely. Last study. This one here, I've been asked before, why do we care? Well, one reason we care is because we use executive control every day in our lives. And I started out by telling you that. And one of the leading causes of death amongst children under the age of 16 is at the crosswalk. It's crossing streets, right? So kids get hit by cars all the time because they don't look both ways, whatever the reason might be. They can't, it's too much information to take in. I'm not really clear. And so we took kids, we put them into our virtual reality environment, and it was a busy crosswalk. And what they did is they were on this treadmill here. Treadmill is not a motorized treadmill. You didn't push a button and it went. You had to actually move it. And so you step forward and it pulled the treadmill back. If you've ever been on a non-motorized treadmill, they're very rare now, but they, prior to motorized treadmills, they used to exist. And so kids would sit there on the treadmill and they would do this task. They'd try and cross the street. So they'd get halfway across, they'd wait, car would go, they'd go again. They just had to walk through that tunnel. They had to do it a couple of times. In the first condition, they just performed the task. In the second condition, we had them do it with a pair of earphones on. They got to select music beforehand. They're listening to music of their choice. And the third condition, we gave them a cell phone. We put a Confederate in the other room, and we had them use a script and have a conversation. We had higher and lower fit kids, and what we found was that higher fit kids outperformed lower fit kids. But similar to the rest of my data, as we upregulated the demands, higher fit kids maintained their accuracy, and lower fit kids performed continuously worse. And so when they're undistracted, just performing the task, higher fit kids are about 10% more accurate. When we had them listen to music, lower fit kids dropped a little bit. But when we gave them that phone, they dropped substantially. And so they were hit by the car more frequently. And if you were to, it's funny here, but of all the things we've done to kids that I've talked to you about today, and things I haven't done, like blood draws and EEG caps and MRI environments, this is the only task that kids didn't like. So this had our largest dropout rate of any other study we did. I don't know why. So in conclusion, fitness may benefit brain health and academic performance. We know that fitness has been linked to changes in cognition, and these changes appear to be disproportionately larger for tasks or task components that require greater amounts of cognitive control. Hopefully, I've convinced you that early physical activity shapes cognition and its neural underpinnings. and that excess adiposity is in some way related to decreased cognition and brain function, but certainly that area needs substantial attention. Single bouts of exercise, I believe, have utility in engendering greater amounts of cognition and more efficient brain function, leading to better scholastic performance. What I didn't tell you before is that this is for about 1 hour after the bout of exercise. When we've tested them out, we see the effects subside about an hour out. And so collectively, hopefully these data suggest that time spent engaged in physical activity doesn't detract from academic achievement, and that early intervention is crucial towards lifespan health and function of both brain and cognition. So thank you.